students, in previous lecture you are studying about nucleic acids, DNA and RNA. Now here we are going to study about chromosomes. Chromosomes. Students, it is also derived from the Greek word the chrome means colored and soma means body. Thus these are colored bodies which are firstly observed by Strasburger, it is just not junk food or burger. It is the name of scientist Strasburger in 1875 into the nucleoplasm, and after him Wardier was the biologist who gave the term chromosome in 1888 to the euchromatics, and you can say that euchromatic threads that are present in the nucleoplasm. Now students, at first we are going to diagrammatic structure of chromosome. What is the structure of chromosome and how it appears? After that, we know more about chromosome. This is the outermost single membranous membrane made up of a very particular type of protein that is called pincer, and here the region of primary constriction where the chromosomes are fused with one another to form a central body that is called centromere. Now more than one chromatids may be present in a chromosome, and at the end of each chromosome a spherical body is present that is called satellite. Now this is the outermost covering of the chromosome. After that, here I am going to represent the internal structures of the chromosome. These are the strands of chromosome. Students, the chromosomes are just threads that contain numerous beads, just like a ring of chains. These are the helical structures which represents the pair of nucleotides. This is the region of primary constriction where first of all these chromatids fused with one another. Now here are the chromosome chromonema, that are just beads-like structure, which appears just white, but it is naturally look like a shape of mustard under a electron microscope. These are the double-stranded helical structure of chromosome, and here I draw the chromomeres. The end of the chromomeres is said to be telomere. The term telomere means end and mere means thread-like structure. Now students, here I am going to label the diagram of the chromosome. This is the outermost membrane of certain type of protein known as pellicum. This is the standard thread-like coiled or helical structure known as chromonemata, and these beads-like structure inside the lies on the chromonemata, that are the chromomere. More ultrascopic structure I can define aside of them. You can easily understand what is the chromonemata and what is the chromomere. These threads-like structures are chromomere. Okay, students, and the beads-like structure that located on the chromomeres at a certain distances, that is called chromomere. I hope you will understand very well. Now the matrix, the fluid content that is filled inside the chromosome, that is chromosomal matrix, or you can say that matrix. This is the centromere where both of the chromonemata get fused to form the region of primary constriction. This is the region of primary constriction. And this is the centromere. It is the central region of the chromosome. That's why this is called centromere. This is the region of secondary constriction. And that is the end part of the chromosome, that is called satellite. And the end of the each chromonemata is known as telomere. Thus, students, beside them, the chromomeres also possess the number of cell organelles like mitochondria, circular DNA or genetic materials, many more. The chromosome in itself a genetic material. because the small segments of chromosomes are known as DNA. Now students, I will describe the chromosome, what is its function, what important role it plays in our cell as well as cell cycle, and how it provides the ability to each and every organism to continue its progeny. Now, first of all, Strasburger in the nucleoplasm observed the thread-like color bodies, and he doesn't describe any name to these bodies, and he easily called them chromatic threads, as you all of you study in the part of the nucleus. But Waldeyer in 1888 described about the euchromatic threads. There I was telling you that there are two types of chromatic threads are present into the nucleoplasm. The unwrapped, uncoiled structures are euchromatic, and the coiled, highly coiled, you can say that the criss-cross shaped structures that are present in the nucleoplasm are dark bands that are the heterochromatics. Here students, the euchromatics are the responsible for the formation of chromosomes, and in each and every organism there is different number of chromosomes are present. Let us take here some examples of species and the number of chromosomes present in them. First I take the example of human. Here I name of species, or you can write here species and number of chromosomes. If you are talking about the number of chromosomes in human or man, then there are 46 chromosomes are found in our body, in which 22 pair, it means 44 chromosomes are autosomes, and the last in the pair, it means 23rd pair, is the sex chromosomes that determines the sexes in organism. More about the autosome and sex chromosomes in ahead lectures. Now here I take the most edible coarse grain, or you can say that cereal wheat. There are 42 chromosomes are found in the wheat, and equal number of chromosomes are also found in the another most edible vegetable, that is potato. 42 chromosomes. And if we are talking about the experimental material of father of genetics, Gregor van Mendel, then we found that there are 14 chromosomes are present in Pisum sativum, or you can say that garden pea. Yes student, 14 chromosomes are found in garden pea, or you can say that its botanical name Pisum sativum. Likewise, in onion, that is also daily used vegetable in our kitchens, onion contains 16 chromosomes. Likewise, if you are talking about the king of animals, it means lion, then we find that there are 38 chromosomes are found in lions. Like many more organisms contain different number of chromosomes. If the number get increased due to any kind of abnormalities, then the characteristics of the species get changed. Due to fluctuation in the number of chromosomes, the characteristics of the species get changed. As if you are talking about the number of chromosomes become 45 in spite of 46, what kind of changes take place in the human body? If there is lack of a single chromosomes in human, then it may be infertile. Likewise, if the number of chromosomes get increases, then their gender can be changed as transgender, or you can say that common gender. Many more about the deformities of chromosomes we look ahead. Now let's come on to the topic. This is the outermost protein membrane, that is division. And here two strands which are wrapped together in a helical way or ladder-like structure, that is called chromonemata. On each chromonemata there is beads-like structures are present. These structures are called chromomeres. And where the chromonemata get fused to form the region of primary constriction, that is called centromere. And further, when the chromonemata get fused to form the region of secondary constriction, and the end of the chromosome form a spherical body, that is called satellite. Students, chromosomes are just nothing but more thing, because these are the structures found in the cell that helps to maintain the progeny of each and every kind of species as well as organism. That's why we can say that it helps in the phenomena like begets like. It means the bitch gave birth to the puppies and the cat gave birth to the kittens. Now students, we are also discussing about the types of chromosomes. Types of chromosomes. There are mainly 4 types of chromosomes, but one type of chromosomes may be also present in different kind of species. First I take the specific type of chromosomes, that is acentric. There is one thing more important to understand, that how the characteristics and the types of chromosomes are classified, on what basis, and how their names get changed. Two things is more important. First, that the structures are called chromatids. These structures, the arm of the chromosomes, you can also say, these are called chromatids. What, students? Chromatids. Okay? And this is the central region of the chromosome or central body, that is called centromere. On the basis of presence and absence of the centromere, the name and characteristics of the chromosomes get changed. Look at it. Acentric. As the name reveals that acentric, it means in such type of chromosomes there is no kind of centromere is present. That's why this is called acentric chromosome. You can define it with the help of such a diagrams, that is chromatids, and nothing presence of the centromere. Second, allocentric. As I define the term telo means at the end. When the centromere present at the end of chromatids, then this is called allocentric. This is called telocentric. Yes, students, this is telocentric chromosome. Here, the centromere is present. Centromere is present at the end of any segments of chromatids, and this is chromatic. Next, I take here the metacentric type of chromosome. As the term meta means in between. When the centromere is present in between the chromatids, it means in between the both arm of chromatids, the centromere is just located. The such type of chromosome is said to be metacentric. And this is the arm of chromatids. Next, I take here submetacentric type of chromosome. As the term reveals that sub means the one arm of chromatids is quite smaller in compared to the another arm of chromatids. That's why this is called submetacentric. Yeah, here we can very well notice that the one of the arm is quite smaller than the another arm. That's why this is called submetacentric, because the centromere is present just aside of between arms. That's why this is called submetacentric. And the last one is acrocentric. Acro means a very small subpart of a chromosome. As you can take this chromatids as a subpart, and another part of the chromatids is like this. This is the centromere and that are the chromatids. Thus, students, you observe that on the basis of presence of centromere or absence of centromere, the name of chromatids and their types are classified. Thus, students, the lecture on the chromosomes will be completed. Thanks for joining today's lecture.